Welcome back to the deep dive. Today, we are cracking open a file that honestly feels less like biology and more like a noir thriller. Or maybe a sci-fi script.
Yeah, it really does.
I mean, it's got everything. Identity theft, breaking and entering a disguised enemy, and this slow burn destruction that literally changed the entire world.
It did, and for the medical students tuning in, this is probably one of the highest yield files we've ever opened. We are talking about an agent that rewrites the rules of the game. It violates the central dogma of biology. It integrates itself into your personal hard drive, and once it's there, it never leaves.
Right. We are talking, of course, about retroviruses. Specifically, we're going to spend the lion's share of this deep dive on the Human Immunodeficiency Virus, HIV, and then we'll pivot to its sort of sinister proliferating cousin, HTLV.
Exactly. And to keep us grounded, we are using Murray's Medical Microbiology, 9th edition, specifically Chapter 54 as our structural spine. We're going to walk through that chapter almost page by page today.
And we're also pulling some crucial clinical pearls from Jowett's, Melnick, and Edelberg's Medical Microbiology, just to make sure we cover the pathology angles thoroughly.
Right. Selectively supplementing from Jowett's Chapter 44 where it really matters for your exam.
But if you're studying, have Murray Chapter 54 open. If you're driving or working out, just listen, because we are going to decode how a virus manages to drive the wrong way down a one-way street without getting pulled over.
That is the perfect analogy to start with. The mission of this deep dive is to understand the mechanism. How does a virus reverse the flow of genetic information? How does it park itself permanently in your DNA? And how does that molecular hijacking lead to the clinical devastation of AIDS or leukemia?
Let's start right there with that rule breaker concept. I mean, in Biology 101, they beat the central dogma into our heads. DNA makes RNA, and RNA makes protein. It's a waterfall. It flows one way.
Correct. That is the fundamental flow of life on Earth. DNA is the master blueprint kept in the vault. RNA is the temporary copy sent to the factory. Protein is the product. But retroviruses, they're named retro because they reverse this flow.
They go backwards.
Yeah, they come in with an RNA genome, but they possess a highly unique enzyme called reverse transcriptase.
RT, the magic wand.
It is a magic wand. This enzyme takes the viral RNA and transcribes it back into DNA. It turns the temporary copy back into a master blueprint.
Which is wild when you think about it. It's like taking a slice of cake and somehow chemically reversing it back into the original recipe card.
And not just a recipe card, but a recipe card that gets filed permanently into the library. Because once that viral RNA is converted to DNA, another enzyme called integrase literally splices it into the host cell's chromosome.
It just pastes right in.
Right in. At that point, the virus is no longer just an invader. It's a gene. It is part of you.
And that's the existential horror part we'll get to later, the permanence. Once you're infected, your own cells are now the enemy. But before we get into the mechanics of the crime, let's look at the lineup. Who are these guys? Murray breaks them down into three families, right?
He does. The broader family is Retroviridae. Inside that, we have three subfamilies, but for human medicine, you really only care about two. First, you have the Oncovirinae.
Onco meaning mass or tumor.
Right. These are the tumor viruses. They're associated with leukemias, sarcomas, lymphomas. The key player here for your exams and for global health really is HTLV1, human T-cell lymphotropic virus.
And that one doesn't kill the cell, right?
Exactly. It immortalizes it. It makes the cell divide forever.
Okay, so the Oncovirinae are the builders. They build tumors. What's the second group?
The Lentivirinae. The root word here is lenti, meaning slow. These are the slow viruses.
Slow like a sloth or slow like a time bomb.
Definitely slow like a time bomb with a very long fuse. The key player here is HIV. These viruses are characterized not by building tumors directly, but by causing neurologic and immunosuppressive disease. They kill the most cells eventually.
So Oncovirinae build, Lentivirinae destroy, and the third group?
The Spumavirinae, the foamy viruses.
Foamy.
Yeah, when you put them in a cell culture in a lab, they cause the cells to look vacuolated and foamy. But here is the high-yield exam tip, and honestly, maybe the only thing you need to know about spumoviruses. There is no known human clinical disease associated with them.
I love that. If you see spumavirus on a multiple-choice question, it is almost certainly a distractor. Just cross it out.
Exactly. Don't waste time on it. Focus on the killers.
Okay, so if we were to shrink down to the nanometer level and look at these killers, what are we seeing?
You're seeing enveloped spherical viruses about 80 to 120 nanometers in diameter.
And that envelope, that's not something they build themselves, is it?
No, that's a stolen coat. They acquire the lipid envelope from the host plasma membrane as they bud out of the cell. But, and this is key, they don't just wear the coat as is, they stud it with their own viral glycoproteins.
Think of it like stealing a car but putting your own custom spikes on the tires.
Great way to visualize it. And inside that envelope, you have the core, or the capsid. Now, Murray makes a very big deal about the shape of this core under electron microscopy.
Why does that matter so much?
It matters because it's a classic exam differentiator. If you look at HIV under an electron microscope, the nucleocapsid core is cone-shaped. It looks like a truncated cone or a trapezoid.
Okay, so HIV is a cone. I'd say C for cone, but that doesn't work because HTLV is the other one, right?
Right. Be careful there. HIV is the cone. HTLV, on the other hand, has a C-type morphology, which means it has a central symmetric core. So remember, HIV equals cone, HTLV equals central or symmetric.
Got it. Cone versus central. Now let's crack open that core. What is the virus packing for its journey?
It is packing a diploid genome. That means it carries 2 identical copies of its positive sense RNA.
Why 2? Is it just a backup?
Evolutionarily, yes. Retroviruses are prone to damage and mutation. Having 2 copies allows for a process called recombination during replication, which helps rescue the virus if 1 copy is broken. It's a built-in redundancy system.
That's smart. But it's not just RNA in there. You mentioned tools earlier.
This is critical for mechanism-based reasoning. Some viruses, like polio, just inject their RNA and trust the cell to do the rest. Retroviruses don't trust the host. They bring their own enzymatic toolbox. They pack about 10 to 50 copies of reverse transcriptase, integrase, and protease inside the capsid.
Like a contractor bringing their own hammer and drill because they know the homeowner doesn't have the right ones.
Exactly. Human cells do not make reverse transcriptase. If the virus didn't bring it, it would be dead on arrival. And here is a tiny nuanced detail from Murray that separates the top students from the rest. The virus also carries 2 cellular tRNAs, transfer RNAs, that it stole from the previous host cell.
It steals tRNAs. What for?
It uses them as primers. You can't start building DNA from nothing. You need a starter block. The virus uses these stolen host tRNAs to prime the reverse transcriptase enzyme. It's highly resourceful.
So it uses stolen tools to start the stolen engine. Okay, let's move to the blueprints. Section 2 of our roadmap in Murray is the genome. He distinguishes between simple and complex retroviruses.
Right. Whether you are simple or complex, all retroviruses have 3 main genes. You need to know these 3 acronyms by heart: GAG, POL, and ENV.
GAG, POL, ENV. It sounds like a strange law firm. GAG, POL, and ENV, attorneys at law. Break them down for us.
Let's stick with your car analogy. GAG stands for group-specific antigen. It encodes the structural proteins, the capsid, the matrix, the nucleocapsid. GAG is the chassis, the frame, and the body of the car.
Okay. And POL?
POL stands for polymerase. This encodes the enzymes we just talked about: reverse transcriptase, protease, integrase. POL is the mechanic and the engine components.
And finally, N.
N stands for envelope. It encodes the glycoproteins, the spikes on the outside. N is the custom paint job and the door handles that let you into the garage.
That makes perfect sense.
Yeah.
Chassis, engine, exterior. So that's a simple retrovirus. But HIV and HTLV are complex. What do they add to the blueprint?
They added a management layer. They have GAG, POL, and N, but they also have a suite of accessory regulatory genes. In HIV, the big ones you need to know are TAT, REV, and NEF.
These sound like characters in a heist movie.
They act like it, honestly. TAT is the gas pedal. It's a transactivator. It tells the host machinery, don't just read this DNA a little bit, read it like crazy. It amplifies transcription 1,000-fold. Without TAT, the virus barely replicates.
And REV?
REV is the traffic cop, or maybe the smuggler. It manages the transport of viral mRNA out of the nucleus. It ensures that the long, unspliced RNAs needed for new genomes get out of the nucleus without being cut up by the cell's normal splicing machinery.
And then there's NEF. I always think of NEF as nefarious.
That is a very appropriate association. NEF is a crucial virulence factor. It's the cloaking device. It downregulates CD4 on the surface of the infected cell, but more importantly, it downregulates MHC class I.
MHC class I is the window display that shows the immune system what's happening inside the cell, right?
Exactly. CD8 T cells, the killer cells, scan MHCI to see if a cell is infected. By pulling MHCI inside and hiding it, NEF makes the infected cell invisible to the immune police. It allows the factory to keep running without being bombed.
Devious. I want to zoom in on the NV gene products for a second. The glycoproteins. These are the lollipops on the surface of the virus. Murray goes into detail on their structure.
This is huge for understanding viral entry and why vaccines are so hard. In HIV, the NV gene codes for a big precursor protein called GP160. This gets sliced into two functional parts, GP120 and GP41.
The head and the stick.
Right. GP120 is the head of the lollipop. It's the surface component. It is heavily glycosylated, covered in sugar molecules, which acts as a shield against antibodies. This is the part that actually binds to the receptors on the human cell.
And GP41.
That's the stick, the transmembrane component. It anchors the complex in the viral envelope. But it's not just a stick. It's a spring-loaded harpoon. When the time is right, GP41 shoots out to pierce the host cell and fuse the membranes together.
So GP120 is the hand that grabs the doorknob, and GP41 is the foot that kicks the door open.
Precisely. And just for completion, since we are covering Murray thoroughly, in HTLV, the equivalents are GP46 for the surface and P21 for the transmembrane. But the HIV GP120-GP41 complex is the one you will see on exams 99% of the time.
One last genomic feature before we watch the crime unfold. The LTRs, long terminal repeats.
These are located at the very ends of the genome. Think of them as the handles of the genetic suitcase. They contain the promoters and enhancers, the instructions that tell the host cell where to start reading and where to stop. They are absolutely essential for the integration process. Without LTRs, the DNA can't get spliced into the host chromosome.
Okay, we've surveyed the enemy. We know the vehicle, the engine, and the crew. Now let's watch the heist. Section 3 of Murray is the replication cycle. He describes this as a target-rich environment for pharmacology, which is a great way to think about it. Every step is a potential drug target. Step 1, entry.
This isn't a simple crash into the cell situation. It is a highly specific, multi-step handshake. First, the viral GP120 must bind to a primary receptor. For HIV, that primary receptor is CD4.
CD4, the defining marker of T helper cells.
Yes, but CD4 is also found on macrophages, dendritic cells, and microglial cells in the brain. So HIV targets the generals of the immune army. But, and this is the crucial exam concept, a binding CD4 is not enough. It unlocks the first latch, but the door is still...
You need the co-receptor.
Correct. Binding CD4 causes a shape change in GP120 that reveals a binding site for a chemokine receptor. There are two main flavors of this co-receptor you need to know, CCR5 and CXCR4.
And the virus is picky, right? It doesn't just grab whichever one is close.
It is very picky, and its preference tells us the stage of the disease. When a person is first infected, the virus is almost always M-tropic, meaning macrophage tropic. It prefers the CCR5 receptor. We call this the R5 virus.
So R5 is the transmission virus. It's the one that gets across the mucosa initially.
Exactly. But as the infection drags on for years, the virus mutates inside the host. In about 50% of patients, the virus shifts to become T-tropic, or the X4 virus. It starts using the CXCR4 receptor instead.
What's the clinical significance of that switch?
The X4 virus is much more aggressive at killing T cells directly. The switch from R5 to X4 often correlates with the rapid crash of the immune system and the progression to frank AIDS. It's like the virus switches from stealth mode to kill mode.
So GP120 binds CD4, then it binds CCR5 or CXCR4. Then what happens?
That second binding triggers the harpoon we talked about. GP41 undergoes a massive conformational change. It unfolds, pierces the host cell membrane, and pulls the virus and the cell together. Fusion happens. The viral core is dumped right into the cytoplasm.
And now, step 2, reverse transcription. The central dogma gets flipped.
The reverse transcriptase enzyme kicks into gear. It takes that single-stranded RNA and synthesizes a complementary DNA strand, a cDNA strand. Then it destroys the original RNA and builds a second DNA strand to make a double helix.
I want to pause on the RT enzyme for a second. We always hear that HIV mutates constantly. Is that specifically because of this enzyme?
Yes, entirely. You have to understand how bad this enzyme is at its job compared to our own DNA polymerases. Our human enzymes have a delete key. They have proofreading activity. If they type a wrong letter, they back up, cut it out, and fix it. RT has no delete key. It is a terrible typist.
So how terrible are we talking?
It makes about one error for every 2,000 to 4,000 bases it copies.
Oh.
Since the HIV genome is roughly 9,000 bases long, that means almost every single new virus produced has at least one or two mutations.
And considering the body produces billions of virions a day,
you generate every possible single base mutation of the genome every single day in an infected patient. We call this a quasi-species. It's not one uniform virus, it's a swarm of variants.
which perfectly explains why we can't just use one drug.
Exactly. If you use one drug, the virus likely already has a variant floating around that happens to be resistant to it. That variant survives, replicates, and suddenly your drug is completely useless. It's also why a vaccine is so incredibly hard. The face of the enemy changes every day.
So we have this sloppy double-stranded DNA copy. Step 3 is integration.
The viral DNA is transported into the nucleus. Now, most retroviruses have to wait for the cell to divide because the nuclear membrane dissolves during division. But HIV is special. It has a nuclear localization signal that lets it actively transit through the intact nuclear pore. It can infect resting, non-dividing cells.
That's a superpower. It doesn't have to wait.
It enters the nucleus, and the integrase enzyme takes that viral DNA, snips the host chromosome, and pastes the virus right in.
And this right here is the point of no return.
Yes. Once integrated, the viral DNA is officially called a provirus. It is now a physical part of the host's genetic material. It is as much a part of you as the gene for your eye color. It can sit there latent for decades.
That is terrifying. It's a sleeper agent.
It is. And as long as it sits there and doesn't make protein, the immune system cannot see it. It is invisible to drugs and invisible to T cells. This is the reservoir that prevents us from curing HIV. We can kill the active virus in the blood, but we can't find the silent proviruses hiding in the DNA.
But eventually it wakes up. Step 4, transcription and translation.
The host cell receives a signal, maybe an infection, maybe cellular stress that activates the cell. The host's own RNA polymerase II starts reading the viral DNA as if it were a normal cellular gene. It makes viral mRNA.
This is where our friends Tat and Rev come in.
Right. Tat hits the gas pedal to make massive amounts of RNA. Rev escorts that RNA out of the nucleus. The cell becomes a full-blown virus factory.
Step 5 is assembly and maturation. This is the endgame of the replication cycle.
The viral proteins, GAG, POL, M, and the RNA genomes assemble at the cell surface. They bud off, taking a piece of the host cell membrane with them. But, and this is a critical clinical distinction, the virus that buds off is immature.
What does that mean? Is it not infectious?
Not yet. Under the microscope, it looks like a donut. The GAG and POL proteins are still stuck together in these long, non-functional chains. To become infectious, the viral protease enzyme has to wake up inside the budded virus and start snipping.
It cuts the chains.
It cleaves the polyproteins into their functional parts. When it does that, the capsid rearranges from that donut shape into the classic cone shape we talked about earlier. This process is called maturation.
And this is a massive target for therapy.
Huge. Protease inhibitors. They work by literally jamming the scissors. The virus still buds out of the cell, but it can't mature. It stays as a donut. It's a dud. It bumps into other cells, but it cannot infect them.
So that's the life cycle. From entry to integration to the release of new duds or cones if we aren't treating it. Now let's talk about the cost of all this. Section 4 in Murray is pathogenesis. What does this actually do to the human body?
We need to talk about two key cell types here, the Trojan horse and the conductor.
Let's start with the Trojan horse.
These are the cells of the myeloid lineage, macrophages and dendritic cells. HIV infects them via the CCR5 receptor. But unlike T cells, macrophages don't usually die from the infection right away. They become persistently infected factories.
And because macrophages are mobile,
They travel, they move into the lymph nodes, they move into the lungs, and critically, they cross the blood-brain barrier into the central nervous system. They carry the virus right into the fortress. This is how you get HIV encephalitis and AIDS dementia. The virus hitches a ride inside the macrophage.
but the main event, the thing that ultimately kills the patient, is the T cell depletion, the CD4 crash.
Right. Murray calls CD4 T cells the conductors of the immune orchestra. They don't do the killing themselves, but they tell the B cells to make antibodies. They activate the CD8 killer cells. They activate the macrophages to eat bacteria.
So if you kill the conductor,
The orchestra stops playing. It doesn't matter if you have violins and drums, B cells and CD8 cells just sitting there. Without the conductor, there is no music, no coordinated immune response. You become susceptible to infections that a healthy person would swat away like a fly.
how does HIV actually kill the T cells? I used to think the virus just multiplied inside until the cell burst, like a balloon.
That's direct cytolysis, and it does happen. But surprisingly, it's not the main way cells die. The mechanisms are much more violent and chaotic. One major way is syncytia formation. Yes. Remember, GP120 is expressed on the surface of the infected cell before the virus even buds. It can bind to CD4 on a passing uninfected T cell. The cell membranes fuse, and another T cell sticks, and another. You end up with this giant multinucleated blob of dozens of fused cells. This giant cell, a syncytium, is totally functionless and eventually dies. So one infected cell can drag dozens of healthy, innocent bystanders to their death.
That's brutal. And you mentioned pyroptosis in our prep.
This is a really hot area of research right now. It's inflammatory cell suicide. In resting T cells, the virus enters and tries to replicate, but often fails because the cell isn't active enough. Viral DNA fragments build up in the cytoplasm. The cell senses this foreign DNA as a massive threat and triggers a self-destruct sequence called pyroptosis.
Pyro meaning fire.
Yes. The cell bursts and releases massive inflammatory signals, cytokines, into the surrounding tissue. This inflammation attracts more T cells to the scene to try and help.
Like moths to a flame.
Exactly. The new T cells arrive, they get infected, or they're triggered to suicide by the inflammatory environment, and the cycle just continues. It's a forest fire of inflammation that rapidly depletes the T cell reserve in the lymph nodes.
Let's zoom out from the cellular level to the population level. Section 5. Epidemiology.
We generally know the routes: sexual parenteral, meaning blood or IV drug use, and perinatal, from mom to baby.
A quick high-yield note on sexual transmission, though. It's not all equal.
No, it's not. Anal intercourse is significantly more efficient for transmission than vaginal intercourse. The rectal mucosa is thinner, more fragile, and crucially, it is densely packed with lymphoid tissue. It's basically a direct doorway to the immune system.
And geographically, sub-Saharan Africa remains the epicenter. But I want to touch on the HIV-1 versus HIV-2 distinction. We mentioned it briefly in the taxonomy.
HIV-1 is the global pandemic. It's aggressive, highly transmissible, and responsible for the vast majority of AIDS cases worldwide. HIV-2 is largely restricted to West Africa.
And it's wimpier, right?
It is. It's less transmissible and progresses to AIDS much more slowly. A patient with HIV-2 might actually live a normal lifespan even without aggressive treatment, though standard of care is still to treat it. But it's really important for medical students to know because some of our standard HIV tests, and particularly our NNRTI drugs, don't work well in HIV-2.
Let's talk about the Berlin patient and genetic resistance. This connects right back to the CCR5 receptor.
This is one of the most fascinating stories in modern medicine. There is a genetic mutation called CCR5 delta 32. It's a 32 base pair deletion. If you have this mutation, your cells make a defective CCR5 receptor that never reaches the cell surface.
So the door handle is just missing.
Exactly. If you are homozygous, meaning you inherited the mutation from both mom and dad, you have zero functional CCR5 on your cells. Since the vast majority of transmitted HIV strains need CCR5 to enter the entropic strains, these people are virtually immune to HIV infection.
The virus knocks, but nobody answers.
Right. This mutation is found in about 1% of people of Northern European descent. The Berlin patient, Timothy Ray Brown, had leukemia and HIV. He received a bone marrow transplant to treat his leukemia from a donor who had this delta 32 mutation. The new immune system that grew in his body was completely CCR5 negative. The HIV in his body had nowhere to go, couldn't infect new cells, and was cleared. He was cured.
It's incredible proof of concept that if you block the entry, you beat the virus. Let's walk through the clinical course of a typical untreated patient. Section 6 in Murray, he outlines 3 phases. Phase 1 is acute infection.
This happens about 2 to 4 weeks after exposure. The virus is replicating explosively. The CD4 count takes a sharp initial drop, and the patient feels terrible.
Like the worst flu of my life.
Fever, swollen lymph nodes, sore throat, maybe a maculopapular rash. It looks exactly like mononucleosis.
And doctors miss it.
All the time. They say, it's a virus, go home and rest. But this is when the patient is incredibly infectious because the viral load in their blood is in the millions. Eventually, the immune system, specifically the CD8 killer cells, rallies and knocks the virus down. Symptoms resolve.
And we enter phase 2, clinical latency, the iceberg.
The water looks calm on the surface. The patient is largely asymptomatic. Maybe they have some persistent generalized swollen glands, but they feel fine. This phase can last for 8, 10, even 12 years.
But underwater?
Underwater, the iceberg is grinding against the hull. The virus is replicating massively inside the lymph nodes. The architectural structure of the lymph nodes is slowly being destroyed. The CD4 count in the blood is slowly, steadily drifting down, losing about 50 to 100 cells per microliter every single year.
Until the bottom drops out. Phase 3, AIDS.
The definition here is strict for board exams. You have AIDS if your CD4 count falls below 200 cells per microliter, or if you develop an AIDS-defining illness, regardless of your CD4 count.
These illnesses, the opportunistic infections or OAIs, they are the bread and butter of clinical exams. They tell a story of complete immune failure. Let's pick a few and explain why they happen. Let's start with the lungs. Pneumocystis.
Pneumocystis jirovecii. This is a fungus. Here's the thing, it's everywhere. You and I probably have it in our lungs right now. A healthy immune system keeps it in check without even trying. But when CD4 drops below 200, the jailer is gone. The fungus proliferates wildly, filling the alveoli with a thick, foamy exudate.
And on an X-ray, this looks like
Ground glass opacities. It looks hazy and bilateral. The patient is profoundly hypoxic, starving for air, but when you listen to their lungs with a stethoscope, they might sound surprisingly clear. That dissociation between the severe hypoxia and clear lung sounds is a classic clinical clue.
Let's go to the brain. Toxoplasmosis.
Toxoplasma gondii. This is a parasite. We get it from undercooked meat or cat feces. Again, many of us have latent cysts in our brain tissue right now. But when CD4 counts drop usually below 100 for this one, those cysts reactivate. They form destructive abscesses in the brain tissue.
Ring-enhancing lesions.
That's the buzzword. On an MRI or CT scan with contrast, you see these glowing rings in the basal ganglia or cortex. It causes seizures, severe headaches, focal neurologic deficits.
And what about malignancies? We mentioned Kaposi sarcoma earlier.
Kaposi sarcoma is the most common cancer seen in AIDS. It's caused by a co-infection with HHV-8, human herpes virus 8. It's a tumor of the blood vessel endothelial cells. You see these dark, purple, violaceous plaques or nodules on the skin, often on the nose or inside the mouth.
It looks almost like a dark bruise that just doesn't heal.
Exactly. And because it's vascular, it's very prone to bleeding. It can also happen internally in the lungs or the GI tract.
So acute to latent to AIDS with opportunistic infections. How do we catch this before the OIs kill the patient? Section 7 in Murray covers diagnosis.
The testing algorithm has been completely updated in recent years. We used to do the Old Testament method, an ELISA screening test, and if that was positive, a Western blot to confirm. The Western blot looked for specific viral protein bands.
But that had a major problem, the window period.
Right. It takes the body weeks to make enough antibodies to show up on those older tests.
Yeah.
If you tested someone 2 weeks after a high-risk exposure, they would test negative, even though they were highly contagious and actively infected.
So now we use the New Testament method, the 5th generation tests.
Now we use a combination antigen-antibody immunoassay. We look for the host's HIV antibodies, yes, but we also look directly for the P24 antigen.
P24, that's the main capsid protein?
Yes. The virus makes P24 well before the human body mounts an antibody response. By testing for P24, we can detect the infection much earlier, sometimes within 10 to 14 days. It narrows that dangerous window period significantly.
And once diagnosed, how do we monitor the patient?
You watch 2 dials on the dashboard. First, viral load using RT-PCR. This counts the number of RNA copies in the blood. It tells you the speed of the disease. It's what we use to see if our drugs are actually working. Second, the CD4 count. This tells you the distance to the cliff. It tells you how much immune damage has already been done and whether you need to start prophylactic antibiotics for things like PCP or toxin.
Which brings us to the good news. Section 8, treatment. We don't just watch people die anymore, we treat them.
We do. HAART, highly active antiretroviral therapy. The golden rule of HIV treatment is you never, ever use monotherapy. You always use a cocktail.
Why is that again?
Because of that bad typist RT enzyme. If you use one drug, the virus mutates around it in a matter of weeks. If you use 3 drugs with 3 completely different mechanisms of action, the virus would have to develop 3 specific simultaneous mutations to survive. The statistical probability of that happening is effectively 0.
Let's group the drugs by the life cycle steps we discussed earlier. This is the absolute best way to remember them for exams.
Okay, 1. Entry inhibitors. Stop the handshake. Maraviroc blocks the CCR5 co-receptor, and Fuvertide binds to GP41 and physically blocks it from folding and fusing the membranes.
2. NRTIs, nucleoside reverse transcriptase inhibitors.
These are fake bricks. They look exactly like normal DNA nucleotides, but chemically, they are missing the 3' hydroxyl group. That's the attachment point for the next brick. When RT grabs one of these fake bricks and puts it into the growing DNA chain, the chain terminates immediately. Game over. Examples are tenofovir, emtricitabine, AZT.
3. NNRTIs, non-nucleoside RT inhibitors.
These aren't fake bricks. They're like gum in the gears. They bind directly to the RT enzyme itself away from the active site and twist the enzyme so it can't function. Example here is efavirenz.
4. Integrase inhibitors.
These are the new heavy hitters in modern regimens. They stop the virus from splicing its DNA into your nucleus. If it can't integrate, it can't establish a provirus. They are highly potent and generally well tolerated. Examples are dolutegravir, raltegravir. Notice the pictogravir suffix.
And 5, protease inhibitors.
The scissors jammers. They bind the viral protease, stopping maturation. The virus still buds, but it stays an immature donut. It's a non-infectious dud. Example is darunavir. Look for the nodavir suffix.
And the ultimate goal of combining these is to reach undetectable.
Yes. If the viral load is undetectable in the blood, the patient stays healthy, their CD4 count recovers, and this is a massive public health victory. They cannot transmit the virus to a sexual partner. U equals U. Undetectable equals untransmittable.
We also use these drugs for prevention now, pre-EP.
Pre-exposure prophylaxis, giving healthy, high-risk people a daily pill, usually a combination like tenofovir and emtricitabine. It acts like a shield. If the virus enters the body, the drug is already inside the T cells, waiting to stop reverse transcription before it even really starts.
And PMTCT, prevention of mother-to-child transmission. We give antivirals during pregnancy, during labor, and we recommend C-sections and avoiding breastfeeding if safe formula alternatives exist. It has drastically reduced pediatric HIV.
It's one of our greatest medical triumphs.
Amazing. Okay, we have thoroughly covered the heavyweight champion, HIV. But we promised to talk about the cousin. Section 9 in Murray covers HTLV.
HTLV-1, Human T-cell lymphotropic virus. The name tells you the fundamental difference. HIV is immunodeficiency. It kills T cells. HTLV is lymphotropic. It makes T cells grow and multiply.
It causes leukemia.
Specifically adult T-cell leukemia lymphoma, or ATLL. But the mechanism of oncogenesis is fascinatingly different from other tumor viruses. HTLV doesn't carry a classic viral oncogene. It uses a viral protein called TAX.
Tax, like the IRS.
Sort of, in that it extracts a heavy toll eventually. TAX is a transactivator protein, functionally similar to TAT in HIV, but whereas TAT turns on viral genes, TAX turns on cellular genes. Specifically, it heavily upregulates the genes for IL-2 and the IL-2 receptor.
And IL-2 is basically T-cell food. It's the primary growth factor.
Exactly. So the virus forces the infected T cell to produce its own food and its own mouth to eat it with. It creates an autocrine signaling loop. The T cell constantly stimulates itself to divide and divide and divide.
Yeah.
This unchecked proliferation accumulates mutations over time and eventually leads to a very aggressive cancer.
But the timeline for this is incredibly slow, right?
It is remarkably slow. The latency period is typically 30 to 50 years. Most people are infected as babies through breastfeeding, but they don't develop leukemia until they are 50 or 60 years old.
And if we look at a peripheral blood smear of a patient with ATLL, what do we see? Because this is the classic board question image.
You look for flower cells. These are malignant CD4 T cells with crazy multilobulated nuclei that look like the petals of a flower or a cloverleaf. If you see the term flower cell in a vignette, the answer is HTLV-1.
It also causes a neurologic disease, but it's not like HIV dementia.
No, it causes HAM-TSP. That stands for HTLV-associated myelopathy or tropical spastic paraparesis. It's a demyelinating disease of the spinal cord. The immune system attacks the virus-infected cells in the spinal cord and damages the upper motor neurons as collateral damage. It causes progressive stiffness, weakness, and eventual paralysis in the legs.
And geographically, where are we seeing this?
It's weirdly patchy globally. It is highly endemic in Japan, specifically the southwestern islands, as well as the Caribbean basin and parts of Central Africa and South America.
All right. We've covered a massive amount of ground today. From the molecular bad typing of reverse transcriptase to the Trojan horse macrophages to the clinical tragedy of AIDS and the flower cells of HTLV.
It's a very dense chapter, but the narrative is consistent. Retroviruses invade the very blueprint of life. They become us.
Before we wrap up, I want to leave the listener with something to chew on. We talked about how terrifying it is that these viruses integrate into our DNA and stay there permanently.
Yes.
But here's the twist. If you sequence the human genome, your genome, about 8% of it, is made up of endogenous retroviruses, or HERVs.
That's entirely true. These are ancient retroviruses that infected our primate ancestors millions of years ago. They infected the germline cells, the sperm or egg, and got passed down generation after generation. They are literally fossilized in our DNA. We are all essentially 8% virus.
And they aren't all bad.
No, they aren't. In fact, we likely wouldn't be here without them. Mammalian biology relies on them. The protein syncytin, which is absolutely essential for the formation of the human placenta, it helps cells fuse to create the barrier between mom and baby, is actually a captured viral envelope protein. We took an ancient viral fusion protein and repurposed it to build human life.
That is mind-blowing. We domesticated a retrovirus to make babies.
Biology's wild. It really blurs the line of what is human and what is pathogen.
Okay, let's see if we can solidify the exam points. I'm going to throw some scenarios at you. Learner, you try to answer it before the expert does. Let's make this a rapid-fire recall. First prompt. I'm looking at an electron micrograph. I see a virus with a cone-shaped core. I know it's a retrovirus. Which one is it?
Cone equals HIV. If it were central and symmetric, that would be HTLV.
Next one. I've got a patient who has been exposed to HIV multiple times but remarkably refuses to get infected. I suspect a genetic mutation. Which host receptor is mutated, and which strain of virus is being blocked?
That is the CCR5 delta 32 mutation. It completely blocks the M-tropic, or R5, strains, which are the ones responsible for initial mucosal transmission.
Third, we talked about the bad typist. Which specific viral enzyme is responsible for the high mutation rate of HIV that leads to rapid drug resistance?
Reverse transcriptase, specifically because it lacks 3' to 5' exonuclease proofreading activity. It's a sloppy copier.
Fourth, I'm looking at a blood smear of a 60-year-old man from Japan with an aggressive leukemia. I see lymphocytes with highly lobulated nuclei that look like flowers. What is the causative agent, and what viral protein is driving the growth?
The agent is HTLV1. The viral protein is TAX, which turns on cellular IL-2 production.
Fifth, a patient with untreated HIV comes in coughing. X-ray shows diffuse ground glass opacities. What is the organism, and what is the CD4 threshold for this specific infection?
The organism is the fungus Pneumocystis jirovecii. The threshold for PCP is a CD4 count below 200 cells per microliter.
Last one. I put a patient on a protease inhibitor like darunavir. Explain to me, on a molecular level, what the newly released viruses will look like.
They will look like donuts. The drug prevents the cleavage of the Gag-Col polyprotein, so the virus cannot undergo structural maturation. It remains immature and non-infectious.
Boom. If you nailed those, you are in fantastic shape for the boards. If not, open Murray Chapter 54 and reread the replication and clinical syndrome sections specifically.
And really, don't just skim it. Look at the diagrams of the replication cycle. Understand the flow and where the drugs work.
Thanks for diving deep with us today. Keep those immune systems strong.
Be curious.